In this video, we're going to discuss some general information about animal senses, and then we're going to examine the anatomy and physiology of the human eye in more detail. So the first thing to consider is how you actually detect what's going on in the world around you and also inside your own body. And the answer to this, the initial step, is with sensory receptors and sensations. So sensory receptors include specialized cells that will detect specific stimuli. So these will include interoceptors, which detect stimuli inside the body, and these include receptors that will detect your blood pressure, your blood volume, and the pH of your bloodstream. These are actually involved in homeostasis. So how do you regulate conditions within your body? You have to first be able to detect conditions within your body in order to be able to regulate them. Now these get regulated by negative feedback. Exteroceptors detect stimuli that come from outside the body. And these would be receptors for things like taste, smell, vision, hearing, and equilibrium. And what they do is they inform your central nervous system about the state of the world around you. Chemoreceptors respond to chemicals, and these would be taste, smell, and blood pH. So you're going to find chemoreceptors for both internal and external sensation. Photoreceptors respond to light energy, and this, of course, is going to be from outside the body. You're not going to generate any light inside your body. And this would be vision. Mechanoreceptors are stimulated by mechanical forces. So this would be hearing, gravity, motion, body position. Generally, mechanoreceptors are stimulated by some sort of stretch or movement. Thermoreceptors are stimulated by changes in temperature, and so these are going to be, again, both external and internal. So they are located in the hypothalamus and in the skin. Now, detection occurs when some sort of environmental change stimulates a sensory receptor, but sensation occurs when the nerve impulses arrive at the cerebral cortex of the brain. So is it actually a sensation at the very moment that your sensory receptors start firing messages toward the brain? No, it's not. It's only sensation when those nerve impulses arrive at the cerebral cortex of the brain. Perception is going to occur when the brain interprets the meaning of the stimuli. So sensation and perception are a different thing. Sensation means that you're detecting something that's going on either internally or externally, but perception is recognition. So let's talk about the pathways that lead to the central nervous system. First of all, in the peripheral nervous system, you'll have some sort of stimulus that's detected by a sensory receptor. That sensory receptor will then send nerve impulses along a sensory fiber to either the spinal cord or to the brain. Now, there are several nerves called cranial nerves that have a direct connection with the brain. Most sensory nerves actually route their way through the spinal cord on their way to the brain. Okay, so if we talk about somatic senses, these are the senses with receptors that are associated with the skin, the muscles, the joints, and the viscera. So there are actually three types of somatic sensory receptors. First, proprioceptors, and we'll discuss that in a little bit more detail, cutaneous receptors, and pain receptors. So proprioceptors, I thought this would be a nice picture to put in there, inform the brain of the body's position, and that's a really unusual position to be informing the brain of. But here's how I want you to test your ability to detect proprioception. What I want you to do is to close your eyes and to touch your nose with the index finger of your right hand. So close your eyes and touch your nose. So you succeeded, right? So this means that you have an innate sense of where the body's position is. It's not something that's dependent on sight or hearing or anything else. It's just you know where your limbs are, and it's proprioception and proprioceptors that inform you of that. Okay, so both layers of the skin contain cutaneous receptors. Cutaneous actually means skin. So there are fine touch receptors, which include Meissner's corpuscles and what's called the root hair plexus. And what the root hair plexus does is to allow sensation when hair is touched. So what I want you to do is run one hand kind of up your forearm, not touching the skin, but just touching the hair on the skin. And you'll notice that you can actually feel the sensation even though you're not directly touching your skin, and even though hair itself is actually not alive. Pressure receptors include Pacinian corpuscles, and there are also temperature receptors, which are free nerve endings. So I want to make something else clear here, and it's that the pressure receptors are not going to be for sensation on the very surface of your skin. So if you squeeze your forearm, then you'll notice that you've got the sensation of skin-to-skin contact, which would be your Meissner's corpuscles. But you can also feel the pressure a little bit deeper, and this would be the Pacinian corpuscles. So as far as temperature receptors go, some of them respond to cold, and those are actually more numerous than the ones that respond to heat. And again, some respond to heat. All right, so if we take a look at the position of these receptors in the human skin, let's first get ourselves sort of oriented here. So this is the epidermis, and everything else beneath the epidermis is the dermis. So where you see that sort of dark purple layer with a darker purple line beneath it, that's going to be the boundary of the epidermis. So what do we have here? Free nerve endings extend up into the epidermis, and they're going to be responsible for heat and cold and, unfortunately, pain. Merkel discs are for touch, and these are going to be for fairly light touch. These are Krauss end bulbs, and I think you can tell from the positioning in the skin that this is going to be for a little bit deeper touch or maybe pressure. These are the root hair plexuses, and you can see them bundled around the very bottom of the hair. So again, if something moves the top of the hair, then that motion is going to be transmitted down to the root hair plexus, and that will give you the ability to feel sort of an amplified sensation from moving of the hair. These are Meissner's corpuscles, which we said were for detecting light touch at the surface of the skin. And these are the Pacinian corpuscles, which are responsible for detecting pressure. And so you can see, since they're deeper down in the skin, they're going to be for sort of deeper sensation. These are the Ruffini endings, and these are also responsible for detecting either light or deeper pressure. Now, as far as pain receptors go, these often get called nociceptors. These get stimulated by chemicals that are released when tissue is damaged, and what they're going to do is to alert us to sort of possible danger or possible damage to the skin. There is something that is called referred pain, and this is a little bit more of a complicated story. So referred pain happens when stimulation of pain receptors also gets perceived as pain from the skin. So a classic example of this is if somebody's having a heart attack, they feel pain down the surface of their left arm. Now, the surface of their left arm has absolutely nothing to do with the pain that's going on in the heart. So here's the reason why referred pain happens. So as sensory nerves from different parts of the body come in towards the spinal cord and come in towards the brain, a lot of the time they'll join into the same nerves. So this means that you're sometimes under sort of intense pain circumstances, your mind can confuse intense pain from one area as pain from a different or from multiple areas, because all of these things are joining, all of these sensory neurons are joining, or nerves are joining as they come in towards the brain. So it would be like tributaries of a river joining into one main river. And so how do you tell which water is from which tributary? The answer is you have a harder time detecting that when it's joined the river, and the same thing kind of goes here. I guess that's the analogy that I can use. So here's the example that I used already. Pain originating in the heart is referred to the left arm and left shoulder. Okay, so moving on to our senses of taste and smell, these are called chemical senses because the receptors that are involved they respond to molecules in the food that we eat and in the air that we breathe. And so this means that taste cells and olfactory cells, which have to do with your sense of smell, are classified as chemoreceptors. So let's take a look at what happens with the sense of taste. So taste buds contain chemoreceptors, and they're located primarily in the tongue. You've got different receptors for salty, sour, bitter, sweet, and umami. Now, the map that I've got, or the picture that I've got at the bottom, is a fairly common map of where these different tastes are actually detected. But recently, there's been a lot more evidence to suggest that you actually taste any of those 5 classes of taste all over the tongue, and that it's not necessarily localized to one part of the tongue. That you've got, say, instead of just the sides of the tongue for sour, that you can perceive sour all over the tongue. Now there may be different concentrations in different places, but you don't have sort of a so to speak, a blind spot for sour on the tip of your tongue. You can still detect sour taste there. Okay, umami receptors detect glutamate. And glutamate, the umami receptors, this would be kind of like richness. So, or something being savory. And that's a different type of receptor than it is for sweet, salty, sour, or bitter. So glutamate is present in monosodium glutamate. That should be monosodium glutamate, or MSG. So MSG used to get a really bad rap. It was held to be responsible for high blood pressure. Nowadays, it doesn't have quite so bad a reputation. And I don't think that MSG is something that you necessarily have to fear. By the way, you can actually buy MSG as a spice in the spice section in the grocery store. So, and it's also an additive that's used in a lot of Asian foods, and it gets called vetsin there. Okay, so the sense of smell, 80 to 90% of what we perceive as taste is actually due to smell. So people who are unable to smell, and this is something that's called anosmic, they lose much of their sense of taste. Olfactory cells are chemoreceptors that are located high in the nasal cavity, and what you perceive as an odor is a combination of a lot of different molecules. And so this activates a specific combination of receptor proteins, and the combination of those signals is what your brain interprets as a specific odor. Yeah, so an odor signature is interpreted by the brain. So let's take a look at olfactory cells, their location, and their anatomy. So basically, this is where they're going to be located. If you look at the second label down, it shows you the olfactory bulb, and leading down from that, you see these nerve fibers that constitute the olfactory epithelium. And so this epithelium is what's going to be contacted by odor molecules that are coming in through the nasal cavity. And what happens here is that if you look at the very bottom, you see that there are olfactory cilia of the olfactory cells. And so these odor molecules actually literally bind to the olfactory cells. And once they have bound to those cells, then they'll actually send the nerve impulses up the sensory nerve fibers to what's called the olfactory bulb, and the olfactory bulb will then lead to the olfactory tract, which leads to the appropriate portion of the cerebrum. So what is it that happens to these odor molecules when they're through stimulating the olfactory cells? They actually get absorbed by the olfactory cells. So remember that the next time that you smell something really bad, that actually becomes part of your body. And I've just ruined a lot of life for you, haven't I? So just again, remember that. Next time you smell something really bad, it's become part of your body. So we're going to spend the rest of this PowerPoint talking about the sense of vision, which requires the eyes and the brain to work together. So about 1/3 of the entire cerebral cortex takes part in processing visual information. So we are much more visual creatures than we are anything else. We're dependent on our sense of sight to a greater degree than a lot of animals are. So usually when I ask people in class if you had to pick one of your senses between hearing, smell, and vision, if you could only keep one of those senses, which one would you keep? Most people would actually answer they'd keep their sense of sight. And so if you were able to communicate with a dog and ask a dog which sense it would keep, it might choose smell, because they're a lot more dependent on their sense of smell than we are, and a lot less dependent on vision than what we are. And a greater portion of a dog's cerebral cortex takes part in processing olfactory information versus visual information. So for us, our sense of sight is really important. For other animals, maybe not quite as much. Okay, so the first thing to say is that invertebrate eyes take a lot of different forms. First of all, you've got eye cups, which can distinguish light intensity and direction. And so what you're going to see is pigment cells right here, a cup of pigment cells right here. They're going to be contacted by the photoreceptor cells right here. And once those photoreceptor cells start firing, then they're going to actually send signals to the nerves leading to the brain. Sorry about that. I'm not great with a mouse. Okay, so eye cups, basically these are really primitive, but what they do have the ability to do is just capture at least some light. And so this organism, and it's a flatworm that's this organism, can actually distinguish where light is coming from and how bright that the light actually is. So compound eyes are going to distinguish shapes, and this is sometimes called mosaic visions. And what I want you to notice here is that you've got multiple lenses, like here and here and here. So you don't have just one giant lens like what you've got in our eye. You've got a lot of different lenses, and each lens is actually associated with one photoreceptor cell. Now these lead into a structure called the ommatidium, which joins all of these images and then sends them to the brain, into the nerve leading to the brain. And so there are some insects that have vision that's a whole lot better than ours is. Dragonflies would be a really good example of that. Okay, now this eye is very much like ours. The eyes that are in a squid or an octopus are very close to the way that ours looks. I mean, if you see them in the wild, they actually look like human eyes. They're very similar. So here with this, you've got a single lens. Okay, you've got the lens will focus light onto a retina that's got photoreceptor cells, and the retina joins into a nerve that leads to the brain. So this setup is almost identical to ours. And I've actually been on a couple of science research vessels that were pulling animals up out of the ocean. One of them was a squid, and what was really odd was to see a squid with this bright green, very human eye look in the middle of it, just looking right back at me. So that was a neat experience. Okay, so taking a look at the anatomy and physiology of the eye. First of all, the eye is an elongated sphere. It's about 2.5 inches in diameter, and it's made up of 3 layers. So the outer layer, and we'll see these in more detail in just a minute, the outer layer is the sclera. It's white, so this is basically the whites of your eyes, and it's fibrous. So the joke that I always make at this point is that if it had been an A&P person in charge of the colonials at the Battle of Bunker Hill, he would have hollered, "Don't fire until you see their sclera," and then none of the farmers would have fired, and we'd have lost the war. And we'd all be talking like this right now. Okay, so interior to this layer, I'm sorry, I should say that part of the sclera is the cornea, and so the cornea is made up of transparent fibers, and so the cornea is not its own layer. It's actually part of the sclera that has a slightly different protein makeup than the sclera. Basically, the collagen fibers that make this up are transparent. It's still very strong, but they're oriented differently in such a fashion that that it makes the cornea transparent. So if you mix up the cornea and the sclera, you know it's coming. I'll say that it's cornea hue. Okay, the conjunctiva is a really thin membrane that covers the sclera, and this is that very outermost layer on the eyeball, and this is actually what gets infected if somebody's got pink eye. The choroid layer is actually deep. It's actually, if you were dissecting from the outside of the eye, it would be deep to the eye, but it is more internal than the sclera is. So this is a middle layer, and it's black, basically. It says it's darkly pigmented. It's black, and it's vascular, meaning that it's got a lot of blood vessels in it. Towards the front, the choroid becomes the iris, and so the iris is the colored portion of your eye, and we'll see what the function of that is in just a minute. Well, how about now? It contains smooth muscle that controls the size of the pupil, and the pupil is an opening into the eye. So if so the pupil is the black part of your eye, and it's not black because it's pigmented, it's black because it's actually an opening that goes into the eye. So let's take a look at the, further look at the A&P of the eye. So behind the iris, the choroid thickens into the ciliary body, and that is sort of an anchor point for ligaments that attach to the lens. So the ciliary body contains the ciliary muscle, and it really is able to change the lens shape for near and far vision. So the lens itself divides the eye into 2 compartments. The anterior compartment is in front of the lens, and it's filled with what's called an aqueous humor. which is clear and has just about the same density of water. The posterior compartment is behind the lens, and it contains something that's called vitreous humor, which I think is closer to maybe the consistency of corn syrup. So the third layer of the eye, so we've talked about the sclera and we've talked about the choroid layer, and the third layer is the retina, which is located in the posterior compartment. Now, if you were dissecting the eye, this would be the deepest layer that you would come to, but if you were inside the eyeball, this would be what you would actually see. So this is kind of on the inner surface of the eyeball. So it's located in the posterior compartment, and it's filled with a material called vitreous humor, which I just mentioned. So the retina contains photoreceptor cells. So this is the sensory layer of the eye. And those photoreceptor cells consist of rods and cones. So rods and cones are for black and white vision and color vision respectively. If you want a cheap way of remembering this, C for cones, C for color vision. Okay, the retina has got a region called the fovea centralis, and this is where your sight is the most sensitive because you've got a higher concentration of cone cells packed there. The optic nerve is formed from sensory fibers from the retina. So if we're taking a look at the lens is actually responsible for focusing images on the retina. And so light comes through the cornea initially and continues as light rays pass through the lens and through both the aqueous and the vitreous humors. And so you've got something that's called visual accommodation, and this is basically for viewing close objects. And so what will happen is the lens will actually round up in order to bring a closer object into focus on the retina. This shape of the lens is controlled by the ciliary muscle. Now we're going to see a picture of the eyeball in just a minute, and I think that'll make more sense. So when the ciliary muscle contracts, the lens will round up. It'll round up, and how far it can round up depends on the elasticity of the lens itself. So it actually turns out that as you get a little bit older, as you get into middle age, the lens tends to become less elastic. And what eventually happens there is the lens can't round up quite enough to bring the image of really close objects into focus. And so this is the reason why you'll see people my age at the grocery store. They're trying to look at a label and they're holding it way out at the end of their at their arm's length. And what they're trying to do is actually be able to focus on it. The reason why they can't focus on it close up is because the lens is not flexible enough. So as this says, the elasticity of the lens may decrease with age. Now, if somebody has had a lens replacement surgery, as I've had, the shape of those lenses cannot be changed at all by the ciliary muscle. And so you've got a really fixed vision. So this is the reason why someone who has had lens replacements always has to have reading glasses. Okay, so now we come to the anatomy of the human eye, and the first thing that we're going to take a look at is the layers. So this is the sclera, or the outermost layer, and it's really tough. It is actually an extension of the dura mater of the brain, and so the sclera, again, it's very tough. This is the choroid layer, and it's showing you something that's sort of pinkish, and those blue and red dots are actually blood vessels. So the choroid layer is, if you were to see it in a dissection, it's pigmented actually black. The retina is interior to the choroid layer, and again, this is what contains the rods and the cones, those photoreceptors, those light-sensing cells. These are the retinal blood vessels, and so the retinal blood vessels nourish the retina with blood. Now, there are a couple of different things that I want to mention here. One is that the pattern of retinal blood vessels is as individual as a fingerprint. So sometimes a retina scan is actually used for purposes of security, just the same way that a fingerprint would be. The second thing is there's something that's called diabetic retinopathy. So if you've got damage to the blood vessels, and not just in diabetes, but something that causes widespread damage to the circulatory system, then retinal blood vessels can be damaged as well, and they can be damaged or they can be burst. And of course, without an ample blood supply, rods and cones start dying. So this retinopathy can be the result of the retina getting an insufficient blood supply, and this is why untreated diabetes can lead to blindness. And this is the optic nerve, and you can see that the retina actually joins into, the entire retina actually joins into the optic nerve. This little dip right here is called the fovea centralis, and it is, again, the highest concentration of cones. This is the posterior compartment that's filled with vitreous humor. So the posterior compartment actually makes up the majority of the eyeball. Now, if we turn our attention to the front of the eye, we're going to start out with the cornea. So this, you'll see that the cornea is continuous with the sclera. It's part of the sclera, but again, the proteins in it are arranged in a different arrangement that allows the cornea to be transparent. This is the anterior compartment. It's filled with aqueous humor. This is the iris. And so the iris basically controls the size of the pupil. This is the lens, and again, it's the lens that's responsible for focusing light on the retina. And how does it carry out this focus? So again, the ciliary body contains suspensory ligaments that will tug on the lens and actually change its shape. So here are the functions of the parts of the eye. I'm not going to read those all off. What I would suggest that you do is pause the video for just a second and read through these. Okay, so how is it that you focus a human eye? So if you look at this, the suspensory ligaments are tightened, okay, and this makes the lens flattened, and this allows you to focus on a distant object. In this, when the suspensory ligaments relax, the lens will round up, and this happens when the ciliary muscle contracts. And this allows you to focus on a near object. And again, if the lens itself becomes less flexible, then it won't round up as much when you're trying to focus on a near object. So what's our visual pathway to the brain? Well, vision begins once light has focused on photoreceptors in the retina, but that's the beginning and not the entirety of vision. So you get some integration occurring in the retina, and when we take a closer look at the retina, I'll explain what that means, where nerve impulses begin. So then the optic nerve transmits the integrated impulses to the brain. So what is the function of photoreceptors? Rod cells have a visual pigment called rhodopsin, and so rod is kind of an abbreviation of rhodopsin, and they're also rod-shaped. But these are really sensitive to light, and they're important for night vision. They also provide peripheral vision and the perception of motion. So whenever you think that you see something moving, especially out of the corner of your eye, it's going to be the rod cells that are responsible for this. Cone cells are located mostly in the fovea centralis, so that sort of dip in the retina that I showed you earlier. These get activated by brighter light. And so this is the reason why when light gets dim, you lose a lot of shades of color, because the cone cells aren't able to perceive dim light nearly as well as the rods do. So your vision kind of becomes a lot closer to black and white as it gets a lot darker. If your cone cells were as sensitive to light as your rod cells were, then you'd be able to detect every shade of color, no matter how dim the light got, which is kind of an odd thought because that's not the way that anybody's eyes work. So cone cells permit fine detail and color perception, and there are actually 3 different kinds of cones. One cone for blue, one cone for green, and one cone for red. And a lot of monitors actually have RGB signals, and that's red, green, blue. And so a lot of our technology for us actually viewing monitors and TVs and movies and stuff like that they kind of mix blue, green, and red wavelengths, and as you alter the proportions of blue versus green versus red, then you're going to start seeing different shades of different colors. Okay, so again, different combinations of stimulation, different colors. So taking a look at the photoreceptors in the eye, that very first label shows you the membrane of the discs that are in rod cells. So there's a rod cell, and you can see again that they're much more numerous than the cone cells are. So rod cells consist of an outer segment, including these membranes, and these have got ion channels in their plasma membranes. And so basically, these ion channels are responsive to the presence of light, and when they let sodium in, they're going to start communicating an action potential to the inner segment here. And it's the inner segment that's really going to send a nerve impulse to the optic nerve. and they would do so with those synaptic endings. So this is a cone cell again, color vision. This is the cell body of the cone cells, and here's the nucleus of the cone cells. And again, synaptic vesicles. So this is where they communicate the fact that they're picking up some sort of visual stimulus to the optic nerve. So we're going to take a closer look at the structure and function of the retina. One thing that I want to point out here that's encircled is the blind spot. And so the blind spot is basically an area where you can't perceive anything at all. And this is something that you tested for in the senses lab. And the reason why you've got a blind spot is because in this area there are no rods and no cones. So take a closer look at the picture here. You see where the bracket is for retina. You'll see sort of three layers there, three different colors of layers there. And the bottom layer is actually the rods and the cones. And you don't see any of that at all in the blind spot. So the reason why you don't have a hole in your vision consistently is because one eye tends to cover for the other. Okay, so now we actually come to the structure and function of the retina. So looking at this, I've got the choroid already labeled because that's not part of the retina. So again, with this, you've got blood vessels here, so this would be an artery, and you've got blood vessels here. This would be a vein. Okay, and now we're going to take a look at the three different layers of the retina. So this is where light strikes first. This is called the ganglion cell layer. This is where light strikes second. This is called the bipolar cell layer. And this is where light strikes third, and this is the rod cell and cone cell layer. So ironically enough, even though the rod cell and the cone cells are the things that detect the light, light actually has to pass through the ganglion cell layer and through the bipolar cell layer before it can get to the rods and the cones. Now, one thing that I mentioned that I would go into more detail earlier is integration of signal. So what I want you to notice is right here, look here and here and here. See, there's a lot of rods and cones here in this diagram. There are fewer bipolar layers, bipolar cells in this layer than there are rods and cones. And the reason why is if you look at the cytoplasmic extensions, you can see that the extensions of this cell, they actually stretch out and reach more than one rod and one cone. And so with the signal from several rods and cones going to one bipolar cell, it means that you've got some integration and some compression of the signal. Now likewise, if you take a look at the bipolar cells, you can see that each ganglion cell can receive signals from more than one bipolar cell here. And you've got further integration of signal. And then your ganglion cells actually join into the optic nerve proper here. So this would be the optic nerve going off. Okay, so again, this is what we mean by integration of signal, that there are more rods and cones than there are bipolar cells. There are more bipolar cells than there are ganglion cells. And so you're kind of compressing, say, from high definition to standard definition when you go from the rods and cones to the ganglion cell layer. And my guess as to why this happens would be that we don't quite have enough cerebral cortex to really handle all of the information that would come in if each rod and cone cell had its own connection to the optic nerve. For animals with better vision than ours, so an eagle would be a great example of this. If an eagle could read, it would be able to read a newspaper at 100 yards. So from one end zone to another end zone on a football field. So that's some pretty incredible vision. They've got a much greater portion of their cerebral cortex devoted to vision processing, and they've probably got less integration than what we do. Okay. So next, let's talk about the visual pathway to the brain. So the function of the retina goes like this. So rods and cone cells synapse the bipolar cells, which synapse the ganglion cells, and the axons of the ganglion cells become the optic nerve. So again, this is the blind spot, and that is an area where you don't have any rods or cones. So again, let me kind of come back to this. So this area right here, this little area right here, is where you've got rods and cones, and you can see that nothing like that is present here in this area. Nothing like that is present here in this area. Sorry, I'm just not great freehanding the mouse, but I think you can tell what I mean. And that's the reason why we've got a blind spot, because there are no rods and cones in that area where the optic nerve actually exits the retina. And again, that means that there's no vision possible in this area. All right, so let's talk about how signals actually get to the brain. So first of all, you've got the right visual field, which is in green, and you've got the left visual field, which is in red. And so the first thing that I want you to notice is that each optic nerve, both here and here, have each one of these has both a left and a right visual field. Now these nerves will cross at the optic chiasma, and I want you to see what happens here. You'll notice that both red pathways go to this side, so the left visual side actually goes to the right side of the brain, and you'll notice that both right visual fields actually go to the left hemisphere of the brain. Now now these are called the optic tract, and so the right optic tract will contain everything from the left visual field, and the left optic tract will contain everything from the right visual field. So each optic tract carries different information. Okay, as these optic tracts get to the thalamus in the brain, then what the thalamus is going to do is it's going to send this signal to the primary visual area of the occipital lobe. So the signal travels actually all the way back to the back of your brain. And so this is where the brain senses the visual information coming in, and the occipital lobe is also where the processing happens, where you get the recognition of what it is that you're seeing.